Yes, as Corinne said, this is the title of my presentation. Feel free to ask any questions. This is going to be the overview of my talk. So I'm going to, for those of you who don't know, many of you know already about PRIDE and protein exchange. I will give a short introduction about them. I will mention different ways in which the data is being reused at present. Then I will talk about in-house project that is ongoing. It's actually the first time that I'm presenting this. So it's data is unpublished, and as I said, it's work in progress. And then I want to finish talking about the work that we are doing developing open analysis pipelines for proteomics data. So for those of you who don't know about it, PRIDE stands for the Proteomics Identifications Database. PRIDE Archive is the archival part of PRIDE and is a database that stores mass spectrometry proteomics data. It was started by Leonard Martens in 2005, 2004, 2005. So we are now like 13 or 14 years old, and we stored all types of mass spectrometry proteomics experiments. Beta support is available for tandem MS approaches, still the main technology used in the field, but any type of data can now be stored. And maybe you are not aware of this, but I just wanted to show some slides about really the growth of data in PRIDE in the last few years. So this only starts from 2012 when we started protein exchange. And this represents the number of data sets submitted per month. And as you can see, there is a steady growth in the last few years or so. And in July, August, and September, really, we had the record months so far in terms of submitted data sets. In September, there were almost 250 submitted data sets. This is just to give you an idea of the scale of public data that is being made available these days. And this just represents the same chart per year. And you can see already that at the beginning of October, we reached the amount of data sets that were submitted in the whole of 2016. Again, just to give you an idea that really a lot of public data is being made available by the community. And this just represents a chart of the growth of different EBI resources. And now PRIDE, representing proteomics data, is in third position in terms of size of DNA of EBI resources. Of course, we are still an order of magnitude less than the DNA resources, ENA and EGA. But you can see now proteomics is the third. The PRIDE is the third resource in size. I just wanted to highlight also for those of you who don't know about this, that PRIDE has been leading since the beginning the protein exchange consortium of proteomics resources, trying to enable a standard data submission and dissemination pipelines between the main existing proteomics repositories. And at the moment, we are 5 resources. So PRIDE in the UK, then Basel Peptide Atlas in Seattle, and Massive in San Diego in the US. JPOS, the Japanese proteomics repository, joined 2 years ago, and this is actually quite new because IPROX joined as the Chinese proteomics repository, and they joined just like 1 week ago or so. So again, just to make you realize that these 5 resources, we are agreeing on common practices for data submission and dissemination of proteomics data. And this is another way to see the amount of data is available. These statistics are a bit out of date because they are till September the 1st, but you can see at that point there were 7,500 data sets, and almost 90% of them were in PRIDE. But of course, other resources are also growing. And also I wanted to highlight that really the main origin of those data sets are the United States. And of course, most of the data sets come from human, but the main model organisms are represented: mouse, Saccharomyces, Arabidopsis, rat, etc. In total, more than 1,100 tax IDs are represented in protein exchange. Okay, so this is just to give you an introduction about the amount of data is there available. But of course, the point of one of the points of having all that, all those data available is that it can be reused by the scientific community, and this is already happening. This is just to show the number of the volume of data downloads in PRIDE. This is only PRIDE. In the last years, we have not included 2017 yet, but the trend continues, and you can see that the downloads of data in PRIDE in the last few years has really grown, and the last year it was 241 terabytes. Also, the website usage and the unique IPs have also grown. So I want to highlight here some of the most interesting cases, or some of the most interesting applications that people are using this public data for. This is not a comprehensive talk about this. I just again want to highlight the most interesting cases. And in my opinion, these most interesting cases are related to what we call data repurposing. So that means that the individual authors can reanalyze the data that were originally submitted, but considering new hypotheses in mind that were not taken originally into account by the original authors. And there are three main types of this kind of applications: the proteogenomics studies, discovery of new post-translational modifications, and meta-analysis studies. So proteogenomics, I didn't include an introductory slide here, but because I assume that most of you knew already, but this is the use of DNA sequencing efforts, RNA-seq sequencing efforts, ribosec, other types of genomics information to generate the databases that are used in proteomics analysis. And there are a lot of people that are using, instead of generating their own data, they are using public data sets to do this kind of proteomics analysis. And indeed, data and public resources can be used for genome annotation purposes, for instance, for the discovery of short open reading frames or translated London coding RNAs. And this is happening, of course, in human. So, for instance, I want to highlight here three public, these are just some examples actually, but there is the group led by Michael Trest in Madrid, that they have been doing a lot of research in this context. But also I want to highlight the work of the group of Gerben Menscheid in Ghent, that they're using PRIDE data to annotate London coding RNAs and sort open reading frames. And they have two resources that that can be accessed by anyone in the community. These are just the papers in there that highlight those resources. But this is not only happening for human, this is also happening for other organisms, of course, mouse model organisms like mouse, rats, Drosophila, but also other microorganisms like Mycobacterium tuberculosis or Helicobacter pylori. And again, these are some examples of publications that have been recently published. There are others also in bioRxiv, but this again just represent some examples. I find one of these every 3 weeks, 1 month, because this is really becoming quite common to publish this kind of studies. The second type of approach, or the second type of utility of data repurposing, of public data reanalysis, is the discovery of new post-translational modifications. There have been a few publications in the last few years that have been devoted to one particular post-translational modifications that has been described de novo. And in this case, the authors use enriched phospho-proteomics datasets, and these are the three examples that can be found in the literature. One paper is in molecular and cellular proteomics by the Bernard-Kuster group. Another one is in other methods, and the other one is in science. But of course, the publication in science is just a small part of the whole study. But in addition to this, there is really now, I think, a strategy that we have been using in-house to discover new post-translational modifications, and is the use of spectral clustering and the detection of consistently unidentified clusters. So what one can do, and you can do it with your own data sets or individual data sets or the whole of PRIDE, one can use a spectrum clustering algorithm that what it does, it groups spectra based on its similarity. And what we have found, and again, it can be found in both individual datasets or all PRIDE datasets, are not originally identified spectra that cluster together. So, and coming from different datasets potentially. So for us, it's a method to target commonly found and identified spectra. Of course, those unidentified spectra can correspond potentially to peptides containing new post-translational modifications or not detected before, or also to new variant peptides. Of course, if you work in proteomics, you know that in the last couple of years, there has been a hype for the use of open modification searches, tools like the MS-Fragger by Alexei Nesvitsky group, and now it is possible to analyze those clusters of unidentified spectra using these tools. And what it is found is, of course, a lot of delta masses. Some of them can be assigned, but of course, some of them cannot be easily assigned. If you want to read more about this approach, you can read our paper from last year. And again, you can do this with individual data sets, but also with complete data sets. And the last point I wanted to mention here is the use of the application in meta-analysis studies, because of course it's not the same to analyze one individual data set submitted by the author than analyzing a lot of data sets together. And in this point, I just wanted to highlight two studies that were published last year. One in Nature Methods, that what they do is do a meta-analysis of subcellular proteomics, subcellular localization of proteins. And this very nice study that was published this year in Molecular Systems Biology by the Marcotte group, when they integrate more than 9,000 mass spectrometer experiments to build a global map of human protein complexes. Again, they didn't generate the data themselves. They took data that were already available and generated these studies. If you want to know more about this, I just have highlighted some of the possible examples for reuse of public data. We have published a couple of reviews in the last couple of years about this topic. Another obvious thing to reuse data is the generation of spectral libraries. Well, there are many other possible uses. I just wanted to highlight the most obvious ones here, or the most interesting ones here. Okay, so the third section of my talk, I'm going to talk about a project that is going in-house here. Again, is the first time I'm presenting this. Is what we call the definition of a functional human phospho-proteome. For that, as you will see, we are using public data from PRIDE. But before, of course, I have to introduce the idea. So, as you know, there is what we could call the observable human phospho-proteome. This is just a publication that was published a couple of years ago by the Matthias Mann's group, where they detected approximately 50,000 phosphosites. Then there is what we could call the empirical human phospho-proteome, and for that we could use a very popular database, PhosphositePlus. And in this database, at the moment, they count about 220,000 phosphosites. And what we want to answer as an output of this project is how much of this human phospho-proteome is actually functional. So there was a publication from Landry et al. In 2009 where they postulated that 65% of it was non-functional indeed. So in order to do this is a joint project. Again, it's work in progress with our colleagues here from Pedro Beltrao group here in EBI. They're just upstairs. And our idea is to establish a functional relevance of human phospho-related residues. But for that, the first step is to determine the functional, what we could call a reference human phospho-proteome. And what we have done for that is to use public data from PRIDE, in this case, concretely 112 human phospho-enriched data sets. Then you will see, I will explain later, the second step is to construct a functional score for those phosphosites. This is work in progress. And the last step, and this has already started, but is still not very advanced, is to assign the biological process to phosphosites and the validation of the findings that have been found in silico. So in order to do that, what we did is to, again, I will explain it later, we have reanalyzed together 112 phospho-enriched datasets coming from human, again, all public datasets in PRIDE. Before we did that, we did different rounds of benchmarking to be sure that the software settings that we were using, our IT was able to cope with it. And for that, I haven't mentioned, but we use MaxQuant for the analysis for different reasons. I'm not going to explain them in detail here, but it was for us, it had a lot of advantages to use MaxQuant. It had also disadvantages, but we decided to move forward with MaxQuant. And to do the benchmarking, we use this publication from Matthias Mann, the data from this publication from Matthias Mann's group that they published in Cell Reports a couple of years ago. So we started analyzing these data sets. We checked and we found very similar results. Then we perform a reanalysis of groups of samples together, so a small number of data sets together. And then we moved directly to the whole reanalysis exercise. And for that, we use a Windows server because MaxQuant, at least till this year, only was able to run in Windows. Now there is a version that can run in Linux, but we haven't used it yet. I want to highlight that the whole process of analysis took almost 2 months because, again, we have reanalyzed together 112 data sets in PRIDE corresponding to 101 cell lines or tissues. In total, almost 7,000 raw files with more than 5 terabytes. Again, running time about 2 months. And as a result, we have found 120,000 highly confident phosphopeptide identifications. This represents, in our opinion, the largest, or at least to our knowledge, the largest to date MS-based phospho-proteomics atlas generated. And these are just some of the numbers for you. We started with more than 137 million MSMS spectra. And these are just some statistics. More than 22,000 PSNs were found at this FDR cutoff. And again, as phosphopeptides, we had almost 120,000, correspond to 19,000 protein groups and about 13,000 genes. And again, for all this, we have really followed very stringent FDR cutoffs. 1% at the protein level and correspond to much less FDR at the PSN level. We also have filtered by using the ASCOR and the ASCOR to differentiate between different possible locations of phosphorylation in the same peptide. So just we have really started to analyze this data now because the whole process finalized 1 month ago or so. And this is just a very raw comparison with the data in PhosphositePlus. And you can see that in PhosphositePlus, there are, as I mentioned before, 220,000 phosphosites. In this case, we have like 120,000. But of course, the level of really confidence of our results, we think that is really huge. More than 50% of results in PhosphositePlus are supported only by 1 or 2 spectra. In our case, about 50% of the phosphosites that we found are supported by at least 100 spectra. And as you can see here, the proportions are also very, very large for phosphosites that are identified by between 25 and 100 spectra. And we really think that the settings that we used for analysis were very stringent. So we are actually happy with the level of redundancy and confidence that we find. So I'm not going to talk more about this. We have started to do more comparisons with previous studies, but I'm going to start talking about the part that has been done by the group of Pedro Beltrão, by David Ochoa more concretely, and it's the construction of a functional score for those phosphosites. As I mentioned before, we finalized the analysis like 1 month ago, So they needed to start with some data to set up the analysis framework. Then they use the phosphosites described in this paper from Judith Billen group that was published also last year in Nature Methods, because they need some alternative data to start developing the functional scoring. So in this paper, with less confidence that we do, they detected 1,009 phosphorylation sites. Really not much information is known about how the analysis was performed, but in any case, this is not important in this case. What I want to highlight is that David, in this case, he used the phospho-proteome generated in that study, so about 19,000 sites. And as a gold standard, he used the 6,500 node regulatory sites that are reported in PhosphositePlus. And with the training and the gold standard set, he used different features that I'm going to explain to generate a model that at the end resulted in a functional score. So what did David use as features? So he has been using a lot of different features coming from different categories. This is just a summary. He has been using features coming from MS evidence, like obviously spectral counts, localization score, number of studies where a particular phosphorylation is found, protein abundance, neighboring PTNs, etc. He has also used a lot of regulatory features, like information about kinase correlation, because the group from Pedro Beltrao are quite expert in this topic. Then he has used a lot of annotation-related features coming from UniProt, usual things like topology, transit peptides like single peptides, domain, composition, protein disorder, for instance. And then he has also used a lot of features related to sequence, protein length, linear motifs, predictive disorder, secondary structure, etc. Then he has also used structural information coming from resources like Interactome 3D. But also the idea is that he's now using information coming from PDB, like interface membership, mutation stability, and others. And then also the group from Pedro Beltrao, they are quite expert in evolution. And for that reason, they have also been using information about residue conservation, phosphostatus conservation, and other kinds of epistatic effects. So using all this together and using the data coming from that publication originally, David has come up with a model that has now started to use in the actual data generated in the pilot. So this is of course going on, is being refined, but we have the initial results that they look very promising. And at the moment, so this second step of building the functional score, we think that has worked also quite well. And now the three things that remain to do are assigning biological process to the phosphosites, improve biological process assignment, and then of course the point that has already been started with some clear cases is the validation of functional relevant phosphosites that have been detected in the study in silico, and Pedro has a subset group in Heidelberg, wet lab there, and have started to validate functionally these relevant phosphosites. So as I mentioned, this is work in progress, and I hope that we will be able to finish the project in the first. Our idea is to finalize it in the first half of next year, 2018. I hope that it has been useful for you to realize one of the things that one can do with really the huge amount of data that are available now in PRIDE and other resources. And then also related to reuse of public proteomics data, the last point of my talk is to talk about open analysis pipelines for proteomics data. So this is something that we have started to do in February this year, with the idea to develop robust, fully reproducible, and automated proteomics analysis pipelines. We have started with data-dependent acquisitions of proteomics workflows that can be deployed in a cloud environment. And the idea is that in the future, we will, once they're ready, we'll make them publicly available in containers, Docker containers, or other type of containers, and they will be reusable by the scientific community. And our idea is to facilitate or enable data reuse. But of course, they can be used with your own data sets as well. This is a project funded by Elixir. Elixir is the European framework for life science infrastructure, and it's a collaboration with the group of Oliver Korbacher in Tübingen and Martin Eisenacher in Bochum in Germany. So we have already developed different exemplary proteomics data analysis workflows, for instance, for standard identification, standard identification for PTNs, quantification for label-free approaches, or label-based approaches, sorry, like SciLac or TNT, iTrack, quality control related pipelines that are quite important to us, and also versions of quantification approaches, including PTMs. And the idea is that we are making these pipelines connected directly to public proteomics data from PRIDE, again, to facilitate data reuse by the scientific community. I didn't mention that for this, we are using the OpenMS framework. I mentioned before that this is a collaboration with Elixir Germany, and this is obvious why we are using OpenMS for this. But of course, it has other advantages, like, of course, all the modularization that is available in the OpenMS tool sets. They have software that can handle different standard data formats from the PSI, and they can integrate third-party software from others easily. So we are using OpenMS from this. In terms of technical details, I'm not going to mention them, but we are using the EBI Embassy Cloud as a deployment platform for these pilot projects. And all the software that we are using is open source, as I mentioned before. That is to make these pilots deployable in other cloud infrastructures like Amazon or Google. And for that, we are using Galaxy as the workflow management system in the background, and we are using Kubernetes as the orchestration system that makes all work together. And of course, as I mentioned before, we are also using Docker containers for the creation of the containers. So this is I forgot to include a slide saying that this is the first phase of the project is already finished. The pipelines are already available in the cloud. We are then testing internally. This is quite challenging to when you use this for the first time. So I think that we still will need like 2 or 3 months of optimization, and after that we will start making the pipelines available to first to close collaborators, and then hopefully to the community as a whole. And this is also work that we are doing now, also for other proteomics approaches. So on the 1st of December, we just started a 3-year BBSRC grants awarded to do pipelines for DIA approaches. So, for instance, SWAT is the main kind of technique or workflow that to start with in the project, because this is a project in collaboration with the Stoller Center. That is a center that is developing biomarkers using proteomics in the University of Manchester, and these are the KPIs of the project. The idea is to, again, start with SWAT, but later extend to MSE and also to orbit trap-based, thermo-based DIA approaches. And then also we have obtained recently a 4-year work contrast grant to do, among other things, proteogenomics pipelines. And this is will be done in collaboration with Jyoti Choudhary in the Institute of Cancer Research in London. This project will not start formally till mid-2018, but we are already starting. Again, the idea is that when all these pipelines are deployed in the cloud, they can be made available to anyone in the community for being reused. Of course, our idea with all this is that we can disseminate data coming from PRIDE and other protein exchange partners into other popular bioinformatics resources like Ensemble for genomics information, UniProt for protein sequence information, or Expression Atlas and Open Targets for protein expression related information. We have the data, we have the pipelines that, again, everyone will be able to use. So we will have results that we will make available in these resources in a completely open and transparent way. This is the idea. Okay, so just a summary to finalize my talk. I think that it was more or less what I expected, 30 minutes. The amount of public proteomics datasets are really on the rise. Now reliable and widely used infrastructure now exists, right, and protein exchange. There are a lot of possibilities open for the use of this data. I mentioned just a few of them: proteogenomics or the discovery of new post-translational modifications or metadata studies, meta-analysis studies, and then there are a lot of possibilities for the reanalysis of very large public data sets. I have mentioned the project that we are doing in-house to determine the human functional phospho-proteome. Is ongoing, and we have also started to work in open and reproducible analysis pipelines for DDA, but also we are starting now also for data independent acquisition and for proteogenomics. And I really want to acknowledge everyone involved in the writing on this. So the work in the Human Phospho-Proteome project has been done mainly by Andrew Jarnosak, but also by David Ochoa and from Pedro Beltrao's group. And also the work in the pipelines have been mainly done by Matthias Walser, with collaboration with other EBI teams as well. But again, everyone is involved in all project related things. And of course, thanks to our funders and to all data submitters. And I'm open to any questions you may have. Thank you very much.
Thank you. Do we have any questions?
Hello, can you guys hear me?
I believe we have a question from Andrew.
Sorry, I can't hear you now. Yes, it's just you sound a lot of very much in the background, at least to me.
Sorry, I'll try to speak up, and great presentation. My question relates to metadata, right? Of course, any data reuse is dependent on having good quality metadata. And I'm wondering if you could comment on sort of how you manage that. Do you just rely on essentially the original submitters having high quality data, or is there some mechanism of community annotation or re-annotation? Your thoughts are appreciated.
Okay, so for the original submissions, we rely on the data is provided by the submitters, and in some cases it's not enough. So when we want to do this kind of efforts to reanalyze data, like the project that I have explained with the human phospho-proteome, there needs to be a human curation effort in the background. So I didn't mention this. I should have mentioned it actually. But those 112 data sets that were selected manually based on different criteria, and they were also re-annotated in some cases in contact with the authors. So I think this is unavoidable if you want to do with a study that of this kind of magnitude. In what we are trying to move forward, and it's good that you also mentioned community-based annotation, is that what is becoming more and more common is that people are reanalyzing the same data sets over and over. So there are a lot of wasted resources in terms of reannotation of data sets that are lost. So what we have been starting to talk about between different people, like for instance the guys in ProteiomixDB, in Bernard Kutz's group, or the guys in Ghent, us here, the group from Lennart Martens as well, is to build some kind of infrastructure to enable community annotation. We need some resources to do that. We have started, we have an idea about how to do it. We hope that in 2018 we can move this forward. And the idea, of course, is to save everyone's time if we do that. But yes, I hope I was clear.